presented by Caltech.
I'd now like to introduce Gloria Ha. Gloria is currently a junior majoring in chemical engineering. She has done 2 serves, and this past summer she worked with Dr. John Kim.
Hi, my name is Gloria Ha, and today I'll tell you about the work I did this summer in the Kim lab at Johns Hopkins. So to begin, we know that in normal oxygen or normoxic conditions, cells undergo aerobic respiration in order to produce energy. So through aerobic respiration, for every 1 molecule of glucose, you get around 30 ATPs. On the other hand, in low oxygen or hypoxia conditions, cells must rely on glycolysis rather than aerobic respiration, and this only produces about 2 ATP per glucose molecule, so it's much less efficient. In tumor cells, we know that the inner cells are not as exposed to oxygen as the outer cells, and thus these inner cells must undergo glycolysis very efficiently in order to produce ATP and keep rapidly growing and dividing. And in cancer cells, this phenomenon is known as the Warburg effect. So in the Kim lab, Ting Han was imaging yeast. He had tagged phosphofructokinase 2, which is an important glycolytic enzyme, with GFP so that he could see where it was in the cell. He imaged it, and what he saw were these bright puncta in every cell, 1 per cell. So it turned out that by letting the culture settle for too long before he imaged, he had created this semi-hypoxic environment. So these puncta are what we've come to call glycolytic bodies, or G-bodies, and we believe that they're aggregates of glycolytic enzymes that come together during hypoxia in order to make the cell produce ATP through glycolysis more efficiently. Since then, our lab has done a lot of work on G-bodies. One big thing was that Greg in our lab purified G-bodies from yeast and sent it in for mass spectrometry analysis. From there, based on which proteins were enriched in the G-bodies compared to the rest of the cell, he found around 32 proteins that likely make up a G-body. Another thing that was done was a genome-wide screen of almost 6,000 genes in yeast. So we were seeing if you delete a gene, does the cell still form G-bodies? So these are examples of the kinds of images you would expect from such a screen. In normoxic conditions, the cell has diffuse PFK to GFP because it's undergoing aerobic respiration and doesn't need to undergo glycolysis efficiently. Whereas when you subject the cell to hypoxic conditions, in the wild-type cells, which are just the native cells without any gene deletions, you see 1 puncta per cell, so 1 G-body per cell. Whereas when you delete SNF1, which is an AMP kinase, you see this fracturing of G-bodies, or multiple dots per cell. And then in SOD1, in hypoxic conditions, the cells look pretty much like the normoxic ones, indicating that SOD1 is a pretty important gene in forming G-bodies. With these initial screens, the mass spec and the genome-wide screen, my job this summer was to process the initial hits. So first, I wanted to take the genome-wide screen, which returned 124 hits, and go through and repeat those experiments and validate them and narrow it down to a smaller number. Next, with the images I got from this analysis, I wanted to automate the image processing. And then lastly, I wanted to find the orthologs in C. elegans, since we're a worm lab. So for the first aim, which was to validate the screen, I first needed to develop a working protocol, and this ended up taking quite a bit longer than I expected. So this is my general experimental flow through. I start with a frozen stock of the yeast deletion strain, which I then streak out onto a plate. And then I grow the plate for 2 days. And then I pick a colony, I make a starter culture, and then I inoculate it into hypoxic and normoxic conditions. And then the next day, I image to see how G-body formation is affected. The problem with this protocol, so I was using 24-well plates and I put a seal to prevent cross-contamination since it's in the shaker. The problem was that my positive control, which is the wild type, which if you'll remember has 1 G-body per cell, was not performing consistently. So some days I would have no G-bodies in any of the cells, some days I would have fracturing, some days I would have what I wanted. So because my control wasn't consistent, I couldn't trust the rest of my results. So I needed to find a method that would give me consistent control behavior. I decided to move on to 5 ml tubes, since these are more commonly used in yeast culture. And what I ended up seeing was that the cells were forming 1 G-body per cell, but in random samples across strains and across days, I was seeing excessive death and debris, as you can see here. And I think it's likely due to overgrowth of the cultures, but with 124 strains with all different growth rates, it wasn't something I could really control for. So I needed a method that would both give me healthy cells and expected behavior in my controls. I moved on to wells without seals, so back to the 24 well plates, but this time I took off the seal because maybe that was the problem. And it turned out that it was. So I initially put a seal on top of the plate in order to prevent splashing between the wells, but it turned out that the seal was also trapping oxygen inside of the wells. So when I wanted a hypoxic environment, I wasn't getting one. And so when I just took off the seal, I got the expected behavior in the control, and this is what I continue to use for the rest of my experiments. I had 124 initial hits, and for each one of those, I wanted to see if you delete a gene, how many cells have one G-body, no G-bodies, or fracturing. And these are examples of my controls. In normoxic conditions, they should have no G-bodies because they're undergoing aerobic respiration, whereas when you subject them to hypoxia, my wild type is the one G-body control, SNF1 is the fractured control, and SOD1 is the no G-body control. And these are the controls I ran with every experiment to make sure my results were replicable. For each strain, I screened them at least twice under the same imaging conditions, and for any that had inconsistencies between the experiments, I repeated them. My next goal was to figure out how to take these images and figure out how many of each kind of cell there are, how many of each phenotype. Traditionally in our lab, how we do this is someone sits in a dark room and opens the image on like Microsoft Paint and goes cell by cell and makes little dots and keeps track of it. So it's accurate, but it's also super time consuming. So I wanted to make a method that was faster, but also accurate. So the first step in doing that was to figure out where the cells are. Each image I took had anywhere from 200 to 400 cells, and I needed to identify those boundaries. So I used CellStar, which is a MATLAB algorithm for yeast cell bright segmentation. So when you plug in this bright field image, it returns where it thinks all of the cells are. And then the green ones are ones I had to modify in some way, but overall you can see it's pretty accurate. And so I used this to find the boundary coordinates of each cell. Next, for each cell, I wanted to know, is there a G-body? Unfortunately, I couldn't get this automated due to some issues with thresholding, but this is what I ended up doing. I had the computer show me the cell, and then I would say whether I saw one G-body, no G-bodies, or multiple G-bodies. So it was kind of a faster method. And so this is just a video of me doing that. I see one, one, one. This is a positive control, so yeah. So through this method, I could get reasonably accurate data for all of my images. And even though it's not as fast as it would be if I could just have the computer do it all for me, it's still a lot faster than the previous method. So it's a step in the right direction. So with this method, I came up with the numbers of each type of cell in each strain. And this is how I displayed the data this is 2 replicates. You can see these are stacked histograms where the bottom black is the percentage of cells with no granules. The middle light gray is the percentage of cells with fracturing, and then the top dark gray is the percentage of cells with 1 granule. So my controls are on the left here, so you can see the 1 granule control has mostly 1 granules, SNF1 has a lot of fracturing, and SOD1 has no granules. For example, these are representative images of some of the deletion strains. LRP1 looks a lot like the positive control, and if you look over here, it does look a lot like the positive control. And so this would be one strain that I would say, I'm not going to validate this strain because it has a lot of G-body formation. Whereas CAN1 looks a lot like SOD1, and it seems like it's really inhibiting G-body formation, and that's how it looks here. So to recap, we started with 6,000 deletion strains and narrowed it down to 124. And then my job was to validate, and I came up with around 60. And so there were about 9 strains that had no G-bodies in any of the cells, anytime I ever imaged them. And then about 10 with fracturing, and then the rest of them had some degree of G-body formation from 1 to 49% of the cells. And these are some representative images of each class. What I found really interesting was that a lot of these genes that I validated were involved in energy generation, whether they were part of mitochondrial maintenance or ATP synthesis, and a lot of them were also chaperone proteins. For example, ATP12 is an assembly factor that's required for the assembly of the alpha and beta subunits of ATP synthase. So it makes sense that if you don't have them, you can't generate as much energy. And so these kinds of enzymes are the ones we're interested in because we want to know the role of G-bodies in energy generation. And then chaperone proteins also got pulled out of the screen, which are interesting because we think the chaperone proteins are helping enzymes aggregate together into G-bodies. So finally, with the hits from both the mass spectrometry screen and from the genome-wide screen, I wanted to find the orthologs in worms, or the conserved genes with the same function in worms. And I found C. elegans orthologs for most of the mass spec hits, and I'm still in the process of doing that for the genome-wide screen hits. And I made a graphical display to show the conservation. So here the green lines are identical amino acids. The yellow lines are similar amino acids based on polarity, and the red ones are completely different amino acids. And you can see that the conservation is higher in the functional domains, indicating that these are likely orthologous genes. So what do we want to do with these orthologs? We want to see if you silence this gene in C. elegans, do you see the same sort of G-body inhibition behavior? And through doing the same sort of screen in C. elegans, we can see things like where are G-bodies forming, what is the tissue specificity. We can see when they're forming in development, and we can also figure out some signaling pathways, since it's a multicellular and much more complex organism. My mentor, Mindy Clark, is working on translating the whole G-body system into worms, but this is difficult and to get a robust reporter. These are some preliminary images, but she's still working on getting something that works. But once it works, we can test everything in worms. Current work that's going on in the lab, Greg and Nathan in the lab are working on characterizing the biophysical and biochemical aspects of G-bodies, and my mentor is working on translating it to worms. Next summer, when I return to the lab, I would like to implement further automation in my image processing thing, not only so that it's faster, but also so I can see things like what is the average size of a G-body for this deletion strain, and what is the quantum yield so I can measure gene expression. I would also like to find the orthologs for both C. elegans and humans for all of my hits, and to link my hits through possible genetic pathways. And then lastly, my main project will be to implement the system in mammalian cells. We've already seen that G-bodies form in mammalian cells, but we haven't looked at different stages of cancer cell lines, for example, or knocked down certain genes. So that's something I'm really excited to further my project through next summer. I would like to thank my professor, John Kim at Johns Hopkins, for giving me a great project and meeting with me every week and being very supportive. And my mentor, Mindy Clark, and Greg Fuller for teaching me all of the yeast experimentation stuff. And Erin Price at the Integrated Imaging Center for helping me with the confocal microscope. And my associate mentor at Caltech, Professor Rob Phillips, and the Caltech student faculty programs for this opportunity. Thank you.
We'd now like to take any questions.
Yeah.
So I might have missed it. You said you think the chaperones are actually helping these G-bodies form?
That's what someone in our lab is trying to look into now. I just heard about this a couple days ago. I don't know what the current status is.
And then what was for SOD1, is it that superoxide displacement?
Yeah.
So why does knocking that out cause no G-bodies to form?
We're not sure. I think it has something to do with the response to oxidative stress, but yeah.
Okay.
That's one of our lab members, that's his favorite gene, so he's looking into it. Yeah.
Do the cells survive less if you have, can you like miss positive results from your knockouts just because the cells are not living that well?
So do you mean like the cells don't grow so I don't see them? There are like some slow growing strains, and so often those correspond to the ones that don't have G-bodies. But in terms of, I think that doesn't confound our results, I guess. Yeah.
Any other questions? If there's no questions, I'd like to thank you.
Thank you.